what if the secret to curing diseases isn't, building a better microscope?
Right.
But instead, it's convincing an AI to just intelligently hallucinate the right answer based on a blurry photograph.
I mean, it sounds a bit like sci-fi when you put it like that.
But think about it. We've spent decades trying to perfect the hardware of structural biology. But today, in this deep dive, we're looking at a totally radical software shift. We're getting into a groundbreaking Nature Methods paper about a new computational tool called Rocket.
Yeah, and it's a fascinating pivot in how we approach molecular biology as a whole. Because, well, for the last few years, the field has been somewhat fractured, I guess you could say.
Fracture how?
Well, you have the computational side, right? Driven by these machine learning marvels like AlphaFold 2. They generate beautifully sharp atomic models from just sequence data in, like, minutes.
Which is amazing.
It is. But then you have the empirical side, the physical experiments, cryo-EM, cryo-ET, X-ray crystallography. These generate the actual literal ground truth of what a protein looks like in reality.
Okay, let's unpack this bottleneck here. Because the AI gives us this pristine prediction, but it's fundamentally unconstrained by the specific physical reality of the experiment.
Exactly. Because of limited training data, machine learning really struggles with condition-dependent changes.
Right.
It throws an absolute fit when trying to model how ligands alter side chain packing, or how a protein radically shifts its shape when it binds to a drug, for instance.
And conversely, the experimental techniques give us the absolute truth of that specific condition, but, and here's the catch, that truth is often maddeningly blurry.
Oh, incredibly blurry. We're frequently dealing with really low-resolution data, worse than 4 or 5 angstroms.
Which basically means we have a beautifully sharp AI guess on one hand and a perfectly accurate but heavily pixelated experimental reality on the other.
That's the perfect way to visualize it, yeah.
So if I'm a researcher sitting with a, say, 5 angstrom cryo-EM map, my immediate instinct is to just take that sharp AlphaFold model and computationally drag the atoms around until they fit into my blurry map.
Which is what people try to do.
Right. So why hasn't that brute force method worked? Because we already have software like ModelAngelo and Predict Then Build, right?
We do, and those tools are powerful. But they operate with severe structural constraints. ModelAngelo, for instance, it requires a full retraining of a novel architecture. And it simply wasn't trained for maps worse than 4 angstroms.
Oh, wow. So it just hits a wall.
It hits a wall. And then you have Predict and Build, which tries to iterate between predicting a structure and then rebuilding it based on the experimental data. But the prediction and the rebuilding steps are completely decoupled.
Meaning they don't really talk to each other.
Exactly. They can actually work against each other mathematically. It prevents the software from ever converging on a definitive answer.
Wait, but standard refinement software has been around for decades. Like, it just applies geometric restraints and literally pulls atoms through 3D Cartesian coordinates to match the density map. It's sort of like having a slightly dented car and hitting it with a rubber mallet until the panel pops back into place. Why doesn't the mallet work here?
What's fascinating here is exactly where that mallet analogy breaks down. I mean, it works for a small dent, say you're tweaking a single side chain rotamer.
Sure.
But when a protein undergoes a massive structural rearrangement, like a dramatic switch in secondary structure, or a whole domain flipping completely to the other side of a hinge, you cannot just drag those atoms across that physical gap in Cartesian space.
Because they crash into things.
Literally, yes.
Yeah.
The optimization algorithms get violently trapped in local minima on the energy landscape. The computer tries to, move an arm through the protein's torso. It hits a massive steric clash.
Ouch.
Right? The energy penalty skyrockets, and the refinement just completely fails.
So we're stuck. Fine-tuning the actual machine learning architectures to fix this is out of the question, I assume, because it's insanely computationally expensive and data hungry.
Very much so.
So if physical restraints and Cartesian coordinates are a dead end for these huge conformational shifts, you have to stop looking at the output and start manipulating the input.
That is the exact conceptual leap behind Rocket.
Which stands for refining open-fold with crystallographic cryo-EM likelihood targets, by the way. Bit of a mouthful.
A huge mouthful. But since we can't force the atoms into place from the outside without breaking the laws of physics, Rocket optimizes the model from the inside out.
Wow.
It actually goes backward into the AI's processing architecture. Specifically, it operates with an open fold, which is the trainable reimplementation of AlphaFold 2, And this is crucial, it doesn't touch the weights of the neural network itself.
Okay, here's where it gets really interesting for me. It targets the MSA cluster profile, the multiple sequence alignment.
It does.
Can you explain that?
Yeah, so OpenFold uses the MSA cluster profile as its core continuous representation of the protein's evolutionary history. The depth, the diversity, the statistical covariation in this profile, that is the blueprint that dictates the final 3D geometry of the predicted protein.
So Rocket intervenes right at inference time then. It iteratively tweaks the mathematical values of that evolutionary history data being fed into the network.
Exactly.
I want to make sure we are crystal clear on the mechanism here for you listening, because this sounds like it's like giving a painter a new mood board while they're already painting.
That's a great way to put it.
If you subtly change the evolutionary inspiration, the AI naturally hallucinate the exact shape you want. And importantly, it maintains perfect stereochemistry because it's using its own native geometry engine to draw it.
The mood board analogy totally works, but the underlying math is significantly more aggressive.
Yeah.
Rocket basically forces a mathematical dialogue between the sequence data and the experimental data.
How?
First, it runs an initial, completely unmodified structural prediction. Then, it performs a rigid body docking to align that prediction with the experimental data. Let's say the X-ray diffraction intensities or the cryo-EM half maps.
But wait, OpenFold generates static structures. It has absolutely no idea what an electron density map or an X-ray intensity even is.
Yeah.
How on earth are you forcing an experimental likelihood target into a sequence-based machine learning model? Are they just like smashing the two data types together?
Not at all. That's the genius of the engineering here. They've made the experimental likelihood targets completely differentiable.
Meaning?
They calculate the error between the AI's predicted structure and the blurry experimental map, and then they backpropagate that error gradient all the way back up to the OpenFold architecture until it hits that continuous representation of the MSA profile.
Oh, wow. So the math flows backward. It tells the system exactly how to apply like multiplicative and additive adjustments to the evolutionary sequence data.
Yes, precisely.
It slightly tweaks the numbers, generates a new 3D shape, checks if it fits the blurry map better, and then loops.
And it does this in two distinct phases to avoid those local minima we talked about earlier. Phase 1 is highly venturous.
Adventurous math. I love it.
Yeah. Rocket utilizes massive learning rates, effectively making huge mathematical jumps in the MSA profile to search for major structural shifts. It forces the AI to abandon its comfortable default prediction and hunt for radical alternative conformations.
It shakes the whole system up. It's basically yelling, go find a totally different fold.
Exactly. And then, once it identifies a macroscopic shape that actually better fits the experimental likelihood target, it shifts to phase 2.
The fine-tuning phase.
Right. The learning rates drop, and it locks in the precise local geometry. But to make this work smoothly, Rocket implements a really sophisticated weighting trick using PLDDT scores.
Right. Okay, the PLDDT, that's the AI's internal confidence metric, right? It tells you which domains it's super certain about and which flexible loops it's basically just guessing on.
Exactly. Rocket extracts those confidence scores and mathematically converts them into pseudo B-factors.
For the listener, what's a B-factor usually?
Oh, sure. A B-factor in standard structural biology is a measure of how blurry or mobile a specific atom is in the experiment. So by using the AI's confidence as a proxy for structural uncertainty, Rocket weights the alignment during the optimization loop.
Ah, I see.
It anchors the optimization around the high confidence regions while allowing the low confidence regions the flexibility to just swing into new positions guided by the experimental density.
Okay, let's just pause here for a quick 5-minute recap so we don't lose the forest for the trees, because this is dense.
Good idea.
So the bottleneck in structural biology right now is that AI guesses are sharp but often wrong for specific real-world states. And cryo-EM maps are perfectly accurate but often terribly blurry. Traditional software tries to literally drag the AI model into the blurry map, but the atoms get stuck crashing into each other. Rocket bypasses this entirely. It backpropagates the error from the blurry map directly into the AI's evolutionary input data. It mathematically mutates the MSA profile until the AI organically folds the protein into the exact shape seen in the experiment.
A flawless summary. Seriously.
Yeah.
But, you know, the true test of any computational tool isn't the elegance of its theory, right? It's the robustness of its application.
Oh, for sure. Show me the data.
Exactly. The researchers validated Rocket by starting with high-resolution challenges and then systematically degrading the data into the blurry unknown.
Okay, let's start with those high-resolution wins. They ran Rocket through 27 different X-ray crystallographic datasets, right? All under 3 angstroms. And the RMSD, the root mean squared deviation, dropped across the board. The models got significantly more accurate compared to the ground truth.
Yeah, and the most compelling of those high-resolution tests involved ligand-induced loop movements, specifically with the human c-Abl kinase.
Okay, break that down for us.
This is a classic challenge. When a small molecule inhibitor binds to c-Abl, a specific loop snaps into a trapped inactive conformation. The base open fold model, left completely unguided, predicts the standard active state.
Because that's what it was trained on mostly.
Right. But when Rocket integrated the X-ray data, it successfully optimized the MSA profile to force that loop into the trapped conformation.
But honestly, high resolution is kind of the easy mode for structural biology, isn't it? If you have a clean 3-angstrom map, humans and standard algorithms can generally figure it out eventually.
Actually, yes.
The real stress test is when the data goes catastrophically bad. And this leads us to the SLC 19A3 thiamine transporter.
Oh, this is where Rocket demonstrates capabilities that fundamentally alter the landscape.
I was blown away by this part.
So the SLC 19A3 transporter sits in the cell membrane, right? It physically opens and closes to allow thiamine to pass through. The original AlphaFold prediction modeled it in an inward open state.
Facing the inside of the cell.
Exactly. However, the experimental map captured it in an outward open state.
Which is an entirely different mechanical pose, a complete macroscopic flip of the domains, like a huge movement.
Massive. So the researchers took a pristine 3.1 angstrom map of that outward open state and began artificially degrading it to simulate terrible experimental conditions.
Just making it blurrier and blurrier.
Right. They added noise, dropping the resolution to 6 angstroms, then 8 angstroms, and finally all the way to 10 angstroms.
Okay, we really need to explain why 10 angstroms is an absolute nightmare, because at 10 angstroms, you don't have atoms anymore.
Not even close.
You barely have alpha helices. The electron density map is so heavily smoothed out that the peaks and valleys that correspond to atomic coordinates completely vanish. There is literally no mathematical gradient for standard Cartesian refinement software to slide down. It's just a lumpy cylinder.
A lumpy cylinder is exactly what it looks like, which makes what Rocket achieved almost unbelievable. Guided by that practically featureless 10 angstrom blob, Rocket successfully forced the starting model to flip from the inward open state to the correct outward open state.
Unbelievable.
The structural prior gradient derived from the MSA optimization bypassed the lack of density gradients completely. And when they superimposed this 10 angstrom guided model against the original clean 3.1 angstrom data,
it matched.
It maintained a degree of agreement that defied the nominal limits of the data. It was stunning.
So what does this actually mean for you, the listener, or for a researcher running a lab right now? Why care about a 10 angstrom blob?
Because getting a 3 angstrom map is incredibly expensive.
And time-consuming.
Yes.
Okay.
And sometimes biochemically impossible for highly dynamic proteins that just won't sit still in the ice. If you can extract high-resolution, atomically precise mechanistic details from fast, cheap, incredibly blurry 10 angstrom experiments, you instantly accelerate the entire pipeline of biological discovery and drug design.
It's game-changing.
It fundamentally democratizes structural biology. Which brings us to the frontier tasks. The cases where human experts, the absolute best in the world, become the bottleneck because the data is just too ambiguous.
Let's look at that specific data set. The human HAI1 protein at 3.82 angstroms.
Right.
In one specific region, residues 310 to 330, the density map was incredibly noisy. At 3.82 angstroms, side chains are often invisible anyway. The human experts who deposited the original model tried to thread the primary sequence through the backbone density, and they got the sequence register completely wrong.
They misaligned the amino acid to the physical shape.
Exactly.
And honestly, it is an incredibly easy mistake for a human to make. You force an alignment, you introduce slight stereochemical strain, and you don't realize you're off by 2 or 3 residues because it's so blurry.
Right. It looks fine to the eye.
But Rocket, running in a completely automated pipeline, corrected that sequence register, because it uses OpenFold's internal geometry engine. It inherently penalizes the steric clashes that a human might accidentally force. It naturally shifted the backbone to perfectly match the density without violating the laws of physics.
So it actually caught a human error in published literature. That's wild.
Yeah.
But what about when the experiment itself is, lying to you? Cryo-EM is famous for artifacts.
Oh, absolutely. If we connect this to the bigger picture of experimental flaws, the ppm1H Rab8A complex is the perfect case study.
Let's hear it.
This was a 3.4 angstrom cryo-EM map, but it suffered from severe preferred orientation.
Meaning what, exactly?
Meaning the protein particles were all sticking to the microscope grid in the exact same orientation. So the researchers were missing critical angular views from the sides or the top. It resulted in an incomplete, smeared 3D reconstruction.
Oh man, it's like trying to mathematically deduce the shape of a car's trunk when you only have 1,000 photos of its front bumper.
That's a really good analogy. And predictably, the standard refinement tools completely failed to orient a vital interaction interface called the flap domain.
But Rocket figured it out.
Rocket cut right through the artifact. It properly positioned the flap domain, revealing crucial structural clues about how this protein specifically selects its substrates. Clues that, remarkably, align perfectly with independent biochemical assays. It extracted deep, actionable biological meaning from profoundly incomplete data.
That is so cool. But for me, the absolute mic drop moment of this Nature Methods paper, the one that really sold me on this whole paradigm, is the ZPD filament validation.
Ah, the avian egg coat protein.
Yes. ZPD forms these long, really complex filaments, and the researchers only had a highly blurry 8.6 angstrom map of this filament. At 8.6 angstroms, you can barely figure out the overall topology, let alone the intricate domain interaction.
Right, it's a mess.
So they ran it through Rocket. And Rocket hallucinates this incredibly specific model where the ZPN and ZPC domains interlock in a very precise way. At the time, there was absolutely no way to prove Rocket was right. It was just a highly educated guess based on a blurry blob.
But science eventually provides the receipts.
Exactly. Later on, an entirely independent research team managed to produce a vastly superior, much sharper 4.6 angstrom map of the exact same ZPD filament.
And when they compared Rocket's speculative guess against the new high-resolution ground truth,
Rocket was completely validated. It had accurately identified the intricate loop interactions stabilizing the filament. It correctly defined secondary structures that were utterly invisible in the 8.6 angstrom map. It saw the truth through the fog, before the fog ever lifted.
it proved definitively that the evolutionary data locked inside the AI's neural weights, when gently perturbed by real-world density targets, can accurately predict physical reality.
So as we hit the final stretch here, let's just look at the big picture. The era of machine learning and empirical experimental data operating in separate silos is ending.
It's over.
Rocket bridges the gap. We are no longer dragging atoms with a rubber mallet. We are mathematically tweaking evolutionary priors to force the AI to generate structures that fit our blurry reality.
Beautifully said.
But, as with all deep dives, we have to look at the boundaries. Nothing is perfect. Where does this tool fail?
Well, the limitations are largely computational and context-specific right now. Calculating those gradients backward through the entire OpenFold architecture is incredibly memory-intensive.
Like, how intensive?
A top-tier 40-gigabyte GPU maxes out at around 500 amino acid residues. So if you are working with massive macromolecular complexes, you have to break the optimization down and process it piecemeal.
You can't just throw a whole ribosome at it yet.
Not without a supercomputer, no. Furthermore, it currently struggles with certain lattice-constrained crystal contacts.
X-ray crystallography issues.
Right. X-ray crystallography forces proteins into unnatural, tightly packed repeating grids. The AI isn't explicitly aware of those artificial intermolecular packing forces, so its gradients can sometimes conflict with the crystallographic reality.
Makes sense.
And lastly, it occasionally fails to flip very small, tight loops, say 3 to 4 residues long, if they have bulky side chains, simply because the energy barrier to move them is disproportionately high in the AI's internal metrics.
Okay, so it's a massive leap forward, but it's a tool, not magic. You still need human intuition to visually inspect the final models.
Oh, absolutely.
But the sheer capability of Rocket raises a philosophical question that I haven't been able to stop thinking about since I read the paper.
It raises an important question indeed. The fact that Rocket works at all is a revelation. I mean, if we can make an AI predict entirely new, completely valid physical conformations, like flipping a massive membrane transporter inside out just by slightly altering the evolutionary sequence data it was fed,
Right.
what does that actually say about the fundamental nature of proteins?
That's the core of it. How much of a protein's physical flexibility, its ability to shapeshift and breathe in a cellular environment, is permanently encoded in its evolutionary history?
It suggests that alternative conformations aren't random accidents of physics. They're deeply conserved evolutionary traits mathematically hidden within the sequence alignment, just waiting for the right computational gradient to expose them.
And that leaves you, listening right now, with a final thought to mull over. If tweaking the evolutionary memory of an AI reveals the secret, hidden poses a protein can strike in real life. Does evolution already know every single shape a protein will ever need to take?
It's wild to think about.
Are those alternative disease-causing or disease-curing shapes completely written into the genetic code, just waiting to be unlocked? Next time you look at a static 3D model of a molecule, ask yourself, what other hidden shapes are locked inside its multi-million-year history, just waiting for the right trigger to reveal themselves? We are no longer just taking pictures of biology, we are interrogating its past to predict its future.